Biology Part 187. Transport of sugars. The products of photosynthesis are transported in the form of dissolved sugars, mainly sucrose. This substance is transported by the phloem from its site of manufacture or storage to the areas where it is used. Any region of supply is referred to as a source, while a region of utilization is called a sink. The leaves of a plant usually act as a source of sugars, and the meristematic growing tissues act as a sink. Storage tissues in structures such as seeds, bulbs, or tubers may be sink during the development and source when being used. The direct transport is generally downward but depends on the seasonal requirement of different plant structures. A possible mechanism of sucrose transport is suggested by the mass flow hypothesis. According to this hypothesis, there are two sites of active transport sugars, both involving companion cells as shown in the figure. At the source, sucrose is taken up actively by the phloem. Companion cells add and passed through the plasmodesmata into the adjacent sieves tubes. At the sink, the reverse occurs, the companion cells actively pumping out sugar into the tissues which require it. This establishes a concentration of gradients of sugars along sieve tube from source concentrated to sink less concentrated. Water is drawn into the sieve tube by osmosis at the source, where the concentration of the sugar is high, and it accumulates the pressure potential and develops into tube, resulting in the bulk movement of the sugar and water towards the sink. At the sink end, the concentration of the sugar in the sieve tube decreases and the water passes out into the neighboring tissues. Among a number of important objections to the mass flow hypothesis is observation that different substances transport in the phloem or sometimes move simultaneously in opposite direction and at different rates. Experimental investigation of plant transport. Radioactive tracers. The site and rate of transport of certain substances can be determined using radioactively labeled plant substrates. The radioactive isotopic phosphorus P, for example, can be incorporated into phosphate and given to the plant in its soil water. Another common substance is carbon dioxide, which can be labeled with furan C and will be absorbed by the leaves of the plant, enclosed in perspex box containing a source of 14CO2. The path of these substances can later be traced by the technique of autoradiography. After drawing and mounting, the whole plant may be placed in contact with X-ray film, which reveals the regions of the highest radioactivity. Alternatively, the plant may be freeze-dried and then cut into the sections suitable for light microscopy. These are covered with the photographic emulsions and later developed when regions of radioactivity show up as black dots, indicating the exact location of the labeled substances. As we have seen, the movement of the water of inorganic nutrients and the sugars in the plants depends on a variety of passive and active transport mechanisms. Although different tissues are specialized in quite different ways, it is important to remember that the substance they carry often have interrelated functions. For example, molecules of sucrose made by the photosynthesis in a leaf may be translocated in phloem sieve tubes to the roots, where they provide glucose for the respiration and intermediates for the synthesis of amino acids. Amino acids are carried upward in the xylem and may later be used to make the new protein differentiating cell at the shoot up. at the shoot tip. Thus, the mechanism described in this unit from a coordinated circulatory system which ensures that all parts of the plant receive the substances they require.